The next two videos are about how genes can be turned on, off, up, or down. This is video 12.4 on Operons, a unique regulation system that has evolved to regulate gene expression in prokaryotes. This video will focus on these 4 unit goals. Pause the video if you want to take a closer look. Remember, prokaryotic cells are cells that lack membrane-bound organelles like a nucleus and endoplasmic reticulum. A good example of prokaryotes is bacteria. Eukaryotic cells, like plant and animal cells, do have membrane-bound organelles. In prokaryotes, not having a nucleus creates an interesting situation where transcription and translation are coupled. Let me explain. You can see in this diagram that DNA is this blue strand right here. RNA polymerase molecules are these small white egg-shaped molecules attached to the DNA. So there are 4 RNA polymerases attached to the same DNA at the same time. So transcription is happening 4 times simultaneously. The mRNA is the red line here. So there's 4 mRNAs being made simultaneously. You can also see that each mRNA has many ribosomes attached. What's happening is that before the mRNA is even fully made, the mRNA is being translated into protein, all of these purple bits coming off. That's what coupling is, when transcription and translation are happening at the same time. Transcription-translation coupling only happens in prokaryotes. Coupling is impossible in eukaryotes if you think about it, because transcription happens in the nucleus and translation happens in the cytoplasm, or the rough endoplasmic reticulum. Moreover, in eukaryotes, the RNA has to be modified before translation can begin. Post-transcriptional modifications of capping, tailing, and splicing don't happen in prokaryotes. In biology, where evolution is king, if a process exists, there has to be some advantage to the process. The obvious advantage for prokaryotes coupling transcription and translation is that a whole bunch of proteins can be made really quickly. But the fact that transcription and translation are uncoupled in eukaryotes has to mean that's advantageous too. In eukaryotes, it means that these processes have more opportunities for regulation of gene activity. But why do you need genes to be regulated? Well, at the basic level, you don't need all of the genes to be turned on at full blast all the time. Certain genes make certain proteins that have certain jobs, so if that protein isn't needed, the gene is turned off until the protein is needed. In multicellular organisms, different cells have different jobs. For example, blood cells transport oxygen, and muscle cells are responsible for movement. And each cell has a specialized structure for their specialized function. So specialized cells need different proteins that need to be turned on at different times and at different levels. And all of that different means gene regulation. Okay, back to this prokaryote story. Because of transcription-translation coupling, when a gene is transcribed, it's kind of like turning the gene on to the max. Kind of like if you turned on your car radio and it was at the highest volume. So when regulating gene expression in prokaryotes, the regulation is usually done at the level of transcription and not at translation or anything in between. To understand gene regulation in prokaryotes better, let's think of this scenario. First, imagine bacteria living in our gut. What would those bacteria eat? Well, whatever we eat. If they're living in our gut, they can't leave and get their own food. So then what if the bacteria specifically needed to have tryptophan? Tryptophan is an amino acid, so tryptophan is a building block needed for all proteins. So to get tryptophan, bacteria would need to rely on us eating tryptophan in our diet, or the bacteria would have to make tryptophan themselves. This is our example scenario, turning genes on or off, regulating the genes for making tryptophan. Since bacteria need tryptophan whenever they're making protein, and that pretty much happens all the time, the genes to make tryptophan are normally on. But if we eat something with tryptophan, the bacteria in our gut don't need to make it because, hey, free tryptophan. In the case that tryptophan is in their surroundings, the bacteria turn the tryptophan-making genes off. In bacteria, the genes needed to make tryptophan are in a location called the Trp Operon. So, an operon is a sequence of DNA that has characteristic parts. The main part of an operon is a cluster of genes that code for proteins that all have a related job. How this works is that RNA polymerase only has to bind to one promoter, and then it makes one single long mRNA that can be translated into 5 proteins. That's hugely efficient. Besides the genes, there are other parts of operons related to gene regulation. A good mnemonic is PROG. P stands for promoter, the spot in DNA where RNA polymerase binds. R stands for repressor. A repressor is a protein that can bind right here. If a repressor is bound, then RNA polymerase can't get through to transcribe the genes. O stands for operator. An operator is the piece of DNA where the repressor binds to block RNA polymerase. Finally is G. G stands for genes in the coding part of the operon. So PROG is the way to go. So let's think more specifically about the Trp Operon. So the proteins made by the genes in the Trp Operon are involved in this multi-step chemical reaction to turn this chemical right here into tryptophan using all of these steps. Each of these steps require an enzyme, and each of those enzymes is coded in the Trp Operon. So when bacteria want to make these proteins, these enzymes, when tryptophan is not in the environment and needs to be made. But when would a bacteria not want to make these proteins? When tryptophan is in the environment. It would be wasteful to go through all of these chemical reactions when the desired product is right there in the environment for the taking. The default or normal position for tryptophan is on. So when tryptophan isn't in the environment, RNA polymerase binds to the promoter and scoots along through the coding region, transcribing the RNA that will later be translated and turned into the proteins to make tryptophan. But when tryptophan is in the environment, the repressor and operator come into play to turn off the Trp Operon. You can see the repressor protein in red here, and the repressor has 2 different shapes. The repressor is a separate protein made by a separate gene. For the repressor to bind to the operator, the repressor has to be in the correct shape. This shape right here fits in the operator, but this shape doesn't. For the Trp Operon, normally the Trp repressor is in a non-complementary shape for the operator and won't fit. But when there's a lot of tryptophan, the tryptophan binds to the repressor. The repressor changes shape, so the repressor can now fit into the operator, blocking RNA polymerase from transcribing the genes, and this turns the operon off. When tryptophan is scarce, this tryptophan molecule will be used and incorporated into a protein, changing the shape of the repressor back to a shape that doesn't fit. The repressor will fall off the operator, and the Trp Operon will be turned on again. So the Trp Operon is another type of negative feedback. We've seen negative feedback before in our signaling unit. For example, when enough thyroid hormone is made, the hormone acts on the thyroid gland, hypothalamus, pituitary gland to shut down its own production. Similarly, in this figure, you can see that the end product of a long series of chemical reactions can feed back as an allosteric inhibitor of the first enzyme in the sequence, shutting down its own production. What's new here is that the product, tryptophan, is also feeding back to the level of transcription to shut down the enzymes from being made in the first place. So it's a 2-pronged attack: shut down the enzyme and shut down the making of more enzyme. Hopefully you can see a connection to evolution here. Imagine how much resources bacteria are saving by not making tryptophan when tryptophan is around for the taking for free in the environment. Another type of operon is called the lac operon. Lac is short for lactose. The genes in the lac operon code for proteins that help digest lactose. But lactose isn't around all the time, so why make genes to digest food that's not there? Instead, the lac operon is normally off, but is turned on in the presence of lactose in the environment. Here's the process of digesting lactose. This enzyme, beta-galactosidase, is a big player in lactose digestion and is one of the 3 genes in the lac operon. Since lactose is normally not in the environment, the lac operon is normally turned off. Let's look at prog. Here's P, the promoter, with RNA polymerase bound. You can see the repressor protein with its 2 shapes, and the shape that fits, operator O, is when lactose isn't around. The genes in the operon include the enzyme to break down lactose, beta-galactosidase. That's G for genes. When lactose does come into the cell, however, the bacteria will want to use that energy source. You can see the lactose in green binding to the repressor and changing the shape of the repressor. With lactose bound, the repressor can no longer fit in the operator. So with lactose present, the repressor falls off of the operator. The repressor is no longer blocking RNA polymerase. RNA polymerase is free to transcribe the genes of the lac operon that can then be translated to make these proteins to digest lactose. We've looked at the trp and lac operons, but these are examples. The trp operon is in a family of operons called repressible operons. Repressible operons usually regulate anabolic pathways that build products. But when there's a free outside source of the product, the operon is repressed or turned off. The lac operon is in a family of operons called inducible operons. Inducible operons usually regulate catabolic pathways that break down substrates. Inducible operons are normally off, but when the substrate is around to be broken down, the operon is induced to turn on. So let's recap what we learned in this video. In prokaryotes, transcription and translation are coupled, making protein production really quick. But it's important to regulate genes because different genes need to be turned on at different levels during different times to make different cells or to have cells respond to different environmental conditions. In prokaryotes, the way that we talked about how genes are regulated is with operons. Operons consist of prog, promoters, repressors, operators, and genes. Some operons are inducible, like the lac operon, and are only turned on when needed. Some operons are repressible, like the trp operon, and are only turned off when they're not needed.